Good afternoon, guys, or morning, depending on when you're hearing this lesson. I don't really know. This is lesson 2.17. This is all about the specific immune response. Here we've got a little starter. If you could please have a go at looking at these questions and answering them in full sentences in your notes, that would be fantastic. I'm not going to go over them, so hopefully you should be able to answer them. If you're not sure, ping me an email, ask questions on the Teams chat, whatever you need to do, and we'll get started straight away. So this is lesson 2.17, specific immunity. We're going to be looking at T cells and B cells, which you've probably never heard about, but will be super confident with by the end of the lesson. We're going to be looking at T cells in a process called cell-mediated immunity and B cells in the humoral response. And we're finally going to spend a little bit of time looking at plasma cells and antigenic memory in terms of long-term immunity to certain conditions. So last lesson, we had a look at antigens and the way that the body distinguishes between self, i.e. us, and non-self or pathogen cells using proteins or recognising those proteins, those antigens, on the outside of the cell. Okay. Now, antigens are those that trigger an immune response. And in triggering that immune response, we get some kind of physiological change that means that we're able to respond to that infection and destroy the pathogen. Now, generally, when we're talking about antigens, we result in the production of an antibody. And the antibody is what we learn at GCSE as things that can fight against bacteria. So what we're going to look at today is how we fight against viruses and a little bit more in detail about this bacterial response system. Now, the specific immune response relies on lymphocytes, that white blood cell that we spoke about last lesson, and these are produced in the bone marrow. So all lymphocytes, all white blood cells are made in the bone marrow and they mature elsewhere. Now, because we're talking about the specific immune response, we know that they're ever so slightly slower. Okay, so they're not instant. The white blood cells or not instantaneous. The white blood cells are not just sitting there floating around in your blood waiting for a pathogen that they've never experienced to come by. Okay, the non-specific immune response, things like your skin, always there. Doesn't matter what the weather is or whether you've encountered that pathogen before, but this specific immune response takes a little bit of time. Okay, they're bespoke to that pathogen. They're not just there. Now, the two types of lymphocytes that we're looking at are both made in the bone marrow, as we said, and that is the spongy stuff inside the long bones. Okay, so we're talking about things like your femur, your thigh bone, or your hip bones. Okay, they're the ones that have got quite a large density, large thickness, and they've got this little spongy matter inside. Now, B lymphocytes, also called B cells, are made in the bone marrow and also mature there. Okay, B for bone marrow. They produce antibodies and fight bacterial infections. Okay, lots and lots of Bs floating around for B cells. However, T cells or T lymphocytes, T cells, mature in the thymus, okay, near where the thyroid glands are, around the throat. And they're used for fighting viral infections. So they work inside cells. And unfortunately for you guys, you need to know that there are specific subtypes of each of those cells, which we're going to spend a little bit of time going over in a second. But before we get there, we're going to have a look back again at what macrophages do. So if you want to, pause now, take a look back at your notes from last lesson. Hopefully that will have only been maybe half an hour ago. And if you're not too sure, ask a question. What do macrophages do? How do they use the antigens from a pathogen once they've ingested it? Now, macrophages present those antigens. Okay, remember, if we're looking at the antigens, these little red blobs on the pathogen is engulfed and the antigens become presenting. Okay, antigen presenting cell, the APC. We can also see antigen presenting cells in cells that have been infected by viruses and other cancer cells. Okay, so if your cell is ever so slightly different from you, the body's like, nah, girlfriend, I'm going to engulf, I'm going to digest, and I'm going to present those antigens. These antigen presenting cells are really, really important in helping trigger T cells. Okay? Now, T cells are those specific immune response cells that mature in the thymus, and therefore viral infections. Just going back to what we did in the previous slide. But they can't respond to antigens in the plasma. Okay? Remember, cells that are infected with viruses aren't going to have necessarily antigens shown on them. Okay? They're just normal body cells. Okay, so virally infected cells just look like normal body cells. And that's why they're so good at evading destruction, okay? Because they just look like normal body cells. Nothing has changed. So instead, what we have to do is we have to make sure our T cells can bind to antigens on the antigen presenting cell. Okay? Now, we're going to go into this process in a little bit more detail in a second. I know it's probably confusing right now. But because the T cells have to respond to a cell, C for cell, we call the T cell activation and response cell-mediated immunity. Okay, it relies on activity or the presence of an antigen presenting cell to activate them. Okay, remember this is about T cells, we're not talking about B cells at the moment. And because we've got this cell-mediated immunity, it's a lot less faffy. Okay, this is the first of the specific responses to be activated. So B cells act after T cells do. Now, the T cells are responding specifically to antigens that are on the antigen presenting cell to activate them. Okay, that's the cell-mediated immunity. So I want you to pause now and think about why the receptors are so important to this response. Okay, think about why it's so important that T cells have those receptors on them. Now, as we can see here in this diagram, we've got a phagocyte engulfing a pathogen. Okay, and we're producing an antigen-presenting cell. Okay, we're presenting antigens here. So we're an APC at the moment, antigen-presenting cell. And see if you can figure out what happens from where I've got my dashed line onwards to make those T cells work. Now, hopefully, you should be able to see that the T helper cell or the T cell receptor fits a particular antigen. It is complementary binding. It is specific. And it is a focused response. Okay? Now, in terms of step-by-steps, this is what you need to know. All right? Keywords are in bold. Hopefully, it's not too difficult. You can draw pictures with them if you like. Ask questions if you're not too sure what's going on. But we start off with macrophages ingesting the pathogens. Okay, they're going to do the phagosome. And we're going to attach it to a lysosome. From last lesson, we form a phagolysosome. We digest our cell, digest our pathogen. Okay, and then we're going to form an antigen-presenting cell, the APC. Yeah? So with that antigen-presenting cell, we know that our pathogen or non-self antigens are going to be displayed on the plasma membrane, that cell surface membrane. Now, we've got a certain type of T cell called a T helper cell. Actually, that should be written T and then a subscript, you know, like when you put H2O, a little h. So T helper cells help the process start. They've got a complementary receptor on them. Okay, that complementary receptor binds to antigens on the macrophage. So we form a T helper cell antigen presenting cell complex. Okay. Now what that T helper cell does once it's bound that antigen presenting cell is like, oh my goodness, there's something that shouldn't be here. Oh dear, I've got to respond to it. So what it does is it produces a certain type of chemical called interleukins. And in your exam, they might call them IL, big I, big L, and a number, say like interleukin 2 or interleukin 7, something like that. Okay, especially if the question's got a big paragraph in them. You don't need to worry about the numbers and stuff unless it specifies a specific number. Just know that big I, big L is an abbreviation for interleukins. Now these interleukins, these chemicals, help further division of the T cells. They divide by mitosis. And we know that in mitosis, we're going to produce genetically identical daughter cells. So what we're doing is we're making sure that we've got lots and lots and lots and lots of the same T cells that know that that's the pathogen that we're responding to. And these clones, because they are genetically identical, they are the same, can become one of 3 different types of T cell. They can become helper cells, which we'll look at in a second, cytotoxic, which we'll also look at in a second, or memory cells, which we'll look at in a second. Alternatively, they can help stimulate phagocytosis even further. So they can sort of opsonize again. Remember we looked at opsonins last lesson? They can flag up the damaged cell or the infected cell and go, it's here, come and get it. Now we said we've got 3 different types of T cells. Okay, we've got those T helper cells, T and a little h, and we know that they produce interleukins. And they're there to stimulate mitosis. Okay, they're also able to stimulate B cell production. Okay, and we know from that little introductory slide that B cells produce antibodies. Now, in doing this whole interleukin process, we're going to start acting on the humoral response as well. Remember those B cells? We're looking at the humoral response. Maybe we're trying to fight a bacterial infection as well. But we're also going to attract further T cells to our environment. We're going to try and get more clones of those cells to help fight that infection. Okay, the next subtype is cytotoxic T cells. Okay, generally if something's got the word toxic in it, it's not going to be a good time. And these cytotoxic T cells produce a chemical called perforin. Now perforin sounds like the word perforates, and that's exactly what this chemical does. It basically punches holes in the walls of the pathogen's plasma membrane. And in doing this, it makes the membrane permeable. It means that water can flow in or flow out because of osmosis. You know, we talked about water potential just before Christmas. And it generally works to destroy the cell. And then finally, we've got T memory cells. There's not really a subscript one for these. But the T memory cells there, and this is a key term, are there for immunological memory. They're your immune system's long-term response. And we say that they persist or stay in blood. Now, what they can do is when you get infected with the same pathogen again, they can super rapidly divide if you get reinfected, and they can form those T killer, those cytotoxic T cells to start perforating, producing perforin and destroying all your infected cells. So could you please have a go now at doing some independent research about the cytotoxic T cells? Okay, these produce perforin, which causes perforations in the cell membrane of pathogens and damaged cells. So those could be the ones that are infected. Find out how producing perforin is an effective method of destroying foreign matter, and explain why the response of cytotoxic T cells is effective against viral infections as well as free moving pathogens, so things like bacteria in the blood. I don't have the answers for these, so you're actually going to have to do it yourselves. Pause now and spend 5 to 10 minutes researching and writing coherent scientific answers for these questions. Alright guys, so the next type of cell that we need to look at is the B cells. Okay, the B lymphocytes are responsible for the humoral immunity, and they respond to pathogens in the bloodstream. Generally, we say this is bacteria, but could also include fungal infections as well. Now, the B cells work by producing antibodies. And these antibodies are soluble protein molecules that mean that they can travel in the blood further distances to treat a far-off infection. Okay, remember that the blood isn't the only fluid that we've got in the body though. Okay, we've also got tissue fluid, the solutions that cells bathe in, and the lymph system, which we'll take a look at maybe later on in this module. Now, the B cells occur and act after the cell-mediated response. Now, if we take a look at antibodies in a bit more detail, looking at their structure, they are specialised glycoproteins. And we learnt about this when we looked at the structure of membranes, the structure of cells. Those are proteins with carbohydrate chains. Okay, so they're a complex of polymers. And the scientific name for these antibodies is an immunoglobulin. And in an exam, they might abbreviate it to IG, big I, little g, just to be annoying. Now, these are specific molecules. They can only bind to one type of antigen, and they bind in the lock and key mechanism. Now, the lock and key mechanism we've noticed, or we've heard about before, in enzymes, but we don't really like it for enzymes because it's not technically true. They do the induced fit model. But for these antibodies and antigens, they specifically bind lock into key, antibody into antigen. And it's this variable region at the top of the Y-shaped region of the antigen that is complementary. Okay, it's that bit that binds to the antigen. It's the antigen binding site, surprisingly, and that's about as much as you need to know structurally. Now, we know that the antibodies can respond in 3 different ways. You might have seen this diagram when you were doing GCSE, especially if I taught you. They can work in 3 different ways. Okay? Antibodies can work to neutralise a toxin by surrounding it. They can do the same with the bacterial cell. Okay, so they neutralise, they surround the danger or the toxin and make it a little bit bigger. They can also work as opsonins, so they flag up the cell for phagocytosis, and also for agglutination. Okay, now agglutination is where the antibodies, I'm going to write AB, so the antibodies work to stick pathogens together. Now, in doing this, they make the pathogen clump a little bit more dense, a little bit more visible for the immune system to respond to. Okay, and then again, that just helps to undergo phagocytosis. Now, in the formation of an antibody-antigen complex or an antigen-antibody complex, whichever way you want to say it, we work to inactivate our pathogen. We don't want it to work. Okay, we've spoken about that neutralisation, agglutination and opsonization, and we're going to spend a little bit more time now just going over those in a bit more detail. So agglutination is where your antibody, this white little Y-shaped diagram, binds to 2 or more antigens, and they could be on different cells. Okay, so in this situation, you can see that we've got some E. coli, a bacterial cell, and it's got lots and lots of antigens on the outside. Now, if we've got these antibodies binding to 2 or more antigens in the diagram over here, on the right-hand side, you can see that the antigens then become clumped together. Again, it's more easily and recognisable for phagocytes, means they can engulf that block all in one go. In terms of neutralisation, we say that these antibodies are going to act as antitoxins. Now, remember, those toxins are going to give us symptoms, and we're going to respond to those by wrapping those antibodies around our toxin molecules to stop them being as effective in the body. Okay, we're not focusing in terms of neutralization, we're not focusing on increasing phagocytosis, we're focusing on stopping the harm to cells. Now again, like the last time, could you please have a go at having a look at the diagram and seeing if you can figure out how B cells work. Okay, it looks very similar at the beginning. Looks a little bit more complicated further downstream though. So I want you to think very carefully about this picture. Pause now, see if you can explain. So hopefully you will have noticed from the previous slide that the B cells are those that become the antigen presenting cells. Okay, it was on a slide a few slides back as well. Now these B cells have already got antibodies on their surface which are complementary to antigens on a pathogen. Okay, and because of that, the pathogen is endocytosed, it's taken into that B cell and enables it to become an antigen presenting cell. Okay, now the antigen presenting B cell now binds to those T helper cells. Now remember we spoke about T helper cells working with B cells. This is where it happens. And those T helper cells produce interleukins. And you can see now how they are linked. Now those interleukins produced by the T helper cells stimulate the B cell to divide by mitosis. So is stimulated, and we're talking about the B cell there, not the T helper cell, to divide by mitosis. Now this division by mitosis is a bit like the fact that the body has gone, I'm picking you, you're the cell that I want, you're the B cell that I need right now, and so we are selection, those clones to be produced. Okay, clonal selection. Now those B cell clones can then either differentiate, so they differentiate there, they become specialised, into plasma cells, which we'll look at in a second, or memory cells, which are very similar to the previous T memory cells that we spoke about. And each one of those plasma cells is able to produce antibodies. Okay, it produces a specific antibodies, but because they're all the same type of antibody, we say that they are monoclonal antibodies. And you will have heard that if you did triple at GCSE. So could you please now have a little look, little Google, little research, and complete this table. There's 3 different types of B cells. Can you figure out what their functions are and what they're there to do? Now we've got 3 different types of B cells again. Okay, we've got B effector cells, and those are the ones that are going to produce plasma cells. They're there to secrete antibodies, those fighters, the things that are going to help fight off those infections. Okay, the plasma cells are producing those specific antibodies, but the reason why we need the B effector cells and we need the plasma cells is because those plasma cells are only alive for a few days. They don't last very long. Okay, but while they're only alive for a few days, they can produce up to 2,000 antibodies a second. They're working hard. Okay, and the final ones we've got are those B memory cells. They're immunological memory, and they just sit around in your blood for decades. Okay, they're really, really long acting, and they're going to help reproduce if you get reinfected, and they're going to help produce antibodies really, really quickly. So let's take a look at that immunological memory. If you've got some notes in front of you, got some paper, you might want to sketch the graph. Take a look and see if you can describe, so say what you see, and explain, give reasons for the graph. Okay, look at the shapes. We've got antibody concentration on the y-axis up the side, and we have got time in days across the bottom. So spend a little bit of time now, draw the graph out, and see if you can say what you see and give reasons for those 2 shapes on the graph. Now what you hopefully should have seen is that exposure to antigen A has a delay to around 6 days before the antibodies to A are produced. Okay, this delay is because it takes time to produce the plasma cells. Okay, we went through that long process. It's going to take more than 2 seconds. Okay, that immune response, the B cells are going to have to divide, they're going to have to specialize, all of that stuff before the plasma cells and then the antibodies can be produced. So from day 7, we've got an increase in antibody concentration over time. Okay, that occurs up until maybe day 15, and after that we see a decline, okay, in the number of antibodies that we've got. We can assume, therefore, that at the peak of this point, we're getting a fighting of the infection successfully. Okay, maybe our pathogen numbers are going down. And after that, we don't need as many antibodies anymore, so the number's just going to reduce. Maybe the plasma cells have died. Okay, at day 28 or thereabouts, we're exposing ourselves to antigens A and B again. Now, we've experienced antigen A already, that's what the blue line is. So we get a rapid increase, and you have to say it's a rapid increase. And with this rapid increase, we have got the memory cells proliferating, and they are producing plasma cells, or they're differentiating into plasma cells, and we're going to increase the amount of antibodies produced. Notice as well that the amount of antibodies produced in this secondary response, okay, 2 degrees secondary, that secondary response produces a much more magnified response to the infection. Okay, you're getting a lot more antibodies and it happens a lot quicker. Conversely, with antibodies to pathogen B or antigen B, you'll see down at the bottom that we're getting that similar primary response because we haven't experienced we haven't encountered that pathogen, that antigen before. Okay, so we're still getting that slight delay. You can see there, we've got the delay between 28 and maybe 34 days, another 6-day delay, and we're getting a slow increase in antibodies and then it levels off again. So these secondary responses are basically controlled and regulated by the ability of memory cells to divide and produce plasma cells. And as we said on that previous diagram, it gives us a much faster response because we don't need to identify the antigen. Okay, we're just making sure that memory cell is able to divide and produce the antibodies properly. Now you've got about 10 minutes to have a go at these 2 questions. These are exam style questions and they bring in things that we've learnt this lesson and at the beginning of module 2. So question 1, compare and contrast cell-mediated and humoral responses to pathogens. So compare the similarities and differences and contrast them. Okay, what happens in this but happens in this one, what happens in this but doesn't happen in this. Question 2, plasma cells can produce up to 2,000 antibodies a second. Suggest and explain 3 organelles that might be present in large quantities in the plasma cells. So pause now. You've got 10 minutes, and when you're done, press play again and go through the answers. Compare and contrast the cell-mediated and humoral responses to pathogens. Now, each of these rows is a separate mark. Okay, for 1 mark, you had to say what happened in the cell-mediated response and then what happens in the humoral response, or what is the opposite to it. So the cell-mediated response involves T cells only, but the humoral response involves mostly B cells. No antibodies are used in the cell-mediated response, whereas they are used in the humoral response. The cell-mediated response is the first part of the specific immune response, whereas the humoral response is the secondary one. And the cell-mediated response is effective through responses to cells specifically. So we're producing that perforin, we're punching through the cell membranes. Whereas humoral responses gives its response through bodily fluid components. That's a fancy way of saying the antibodies floating in the blood. If you've got any marks that you think are questionable and you want to find out whether you would have got a mark for it, then please pipe up on our call and have a question, ask. You will not get the mark if you've just said that cell-mediated involves T cells, no antibodies, blah, 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 and you haven't compared it to the other side though. Now, this plasma cells, suggest and explain 3 organelles that might be present in large quantities in plasma cells, is all to do with the fact that those antibodies are proteins. So you need to think very specifically about which organelles are going to be key and super responsible for making vast quantities of proteins. So firstly, you needed to say the rough endoplasmic reticulum. They're there for making and transporting the antibody proteins. The Golgi apparatus is there to modify the antibody proteins, maybe attaching that carbohydrate subunit to it. And then finally, we've got mitochondria that is there to release the energy for massive antibody production. It suggest and explain. It's only 3 marks, so if you haven't told me why for each of those, you don't get any marks. Read the question properly. If you would like to, I know it says homework, but we don't know where we're going to be this time next week. So pause now. See if you can have a go at doing a flow diagram or a set of cartoons showing how the immune response interacts with each other. How does the non-specific immune response lead to the specific immune response? And where do the T and B cell responses combine? I'm going to be quiet now. I'm just going to work through these next few slides. The questions are on there. Pause, and I'll change the slide every few seconds or so. Answer them in full sentences, reading the statements at the top, and then the answers will be at the end, and I'll say goodbye then. Alright guys, so hopefully you found that useful, informative, and if you've got any questions, remember you can email or pop up on Teams and type it into the chat box, or you can just pipe up on the call and ask any questions there as well. Okay, until next time, thanks again, see you later, bye.